read something that I've not done before is a challenge. So this is what I want to do. I want to read this biochemistry textbook from one to end. So it's going to be a big one, right? So I want to do this with you all. I've actually eaten. This is not in my room. I'm in my department lab. I'm graduated. So I came here to do this. Yeah, because of some reasons best known to me. So I want you to, watch this video till end. It will mean a lot to me. I'm doing something that is crazy, right? Reading a book. Who cares? I know you care. So yeah, just join me as I do this, and hopefully it will mean a lot to all of us. Okay. Biochemistry can be defined. This is chapter one. Biochemistry can be defined as the science concerned with the chemical basis of life. The cell is a structural unit of living system. Thus, biochemistry can also be described as a science concerned with the chemical constituents of living cells and with the reactions and processes they undergo. By this definition, biochemistry encompasses large areas of cell biology, of molecular biology, and of molecular genetics. The aim of biochemistry as a heading is to describe and explain in molecular terms all chemical processes of living cells. The major objective of biochemistry is the complete understanding at the molecular level of all the chemical processes associated with living cells. To achieve this objective, biochemists have sought to isolate the numerous molecules found in cells, determine their structure, and analyze how they function. Many techniques have been used for these purposes. Some of them are listed below. Now, a knowledge of biochemistry is essential to all life sciences. The biochemistry of the nucleic acids lies at the heart of genetics. In turn, the use of genetic approaches has been critical for elucidating many areas of biochemistry. Physiology, the study of body function, overlaps with biochemistry almost completely. Immunology employs numerous biochemical techniques, and many immunological approaches have found wide use by biochemists. Pharmacology and pharmacy rest on a sound knowledge of biochemistry and physiology. In particular, most drugs are metabolized by enzyme catalyzed reactions. Poisons act on biochemical reactions or processes. This is the subject matter of toxicology. Biochemical approaches are being used increasingly to study basic aspects of pathology, the study of disease, such as inflammation, cell injury, and cancer. Many workers in microbiology, zoology, and botany employ biochemical approaches almost exclusively. This relationship are not surprising because life as we know it depends on biochemical reaction and processes. In fact, the old barriers among the life sciences are breaking down, and biochemistry is increasingly becoming their common language. A reciprocal relationship between biochemistry and medicine has stimulated mutual advances. The two concerns for workers in the health sciences, and particularly physicians, are the understanding and maintenance of health and the understanding and effective treatment of diseases. Biochemistry impacts enormously on both of these fundamental concerns of medicine. In fact, the interrelationship of biochemistry and medicine is a wide two-way street. Biochemical studies have illuminated many aspects of health and disease, and conversely, the study of various aspects of health and disease has opened up new areas of biochemistry. Some examples of this two-way street are shown below. That's the table that we'll be looking at, hopefully. For instance, a knowledge of protein structure and function was necessary to elucidate the single biochemical difference between normal hemoglobin and sickle cell hemoglobin. On the other hand, analysis of sickle cell hemoglobin has contributed significantly to our understanding of the structure and function of both normal hemoglobin and other proteins. Analogous examples of reciprocal benefits between biochemistry and medicine could also be cited in that same table. Another example is the pioneering work of Archibald Garrod, a physician in England during the early 1900s. He studied patients with a number of relative rare disorders: acaptonuria, albinism, cystinuria, and pentosuria. These are described in the later chapters, yes, and established that these conditions were genetically determined. Garrod designated this condition as inborn errors of metabolism. His insight provided a major foundation for the development of the field of human biochemical genetics. More recent efforts to understand the basis of this genetic disease known as familial hypercholesterolemia, which results in severe atherosclerosis at an early age, have led to dramatic progress in understanding of cell receptors and of mechanisms of uptake of cholesterol into cells. Studies of oncogenes in cancer cells have directed attention to the molecular mechanism involved in the control of normal cell growth. These and many other examples emphasize how the study of disease can open up areas of cell function for basic biochemical research. The relationship between medicine and biochemistry has important implications for the former. As long as medical treatment is firmly grounded in a knowledge of biochemistry and other basic sciences, the practice of medicine will have rational basis that can be adapted to accommodate new knowledge. This contrasts with unorthodox health courts, at least some alternative medicine practices, which are often founded on little more than myth and wishful thinking and generally lack any intellectual basis. Now, as a heading, normal biochemical processes are the basis of health. The World Health Organization, WHO, defines health as a state of complete physical, mental, and social well-being, and not merely the absence of disease and infirmity. From a strictly biochemical viewpoint, health may be considered that situation in which all of the many thousands of intra and extracellular reactions that occurs in the body are proceeding rates commensurate with the organism's maximum survival in the physiologic state. However, this is an extremely reductionist view, and it should be apparent that caring for the health of patients requires not only a wide knowledge of biologic principles, but also of psychological and social principles rather. As you heard, biochemical research has impact on nutrition and preventive medicine. One major prerequisite for the maintenance of health is that there be optimal dietary intake of a number of chemicals. The chief of these are vitamins, certain amino acids, certain fatty acids, various minerals, and water. Because much of the subject matter of both biochemistry and nutrition is concerned with the study of various aspects of these chemicals, there is a close relationship between these two sciences. Moreover, more emphasis is being placed on systematic attempts to maintain health and forestall disease, that is, on preventive medicine. Thus, nutritional approaches to, for example, the prevention of atherosclerosis and cancer are receiving increased emphasis. Understanding nutrition depends on a great extent on the knowledge of biochemistry. Now, as a heading, most and perhaps all disease has a biochemical basis. We believe that most, if not all, diseases are manifestations of abnormalities of molecules, chemical reactions, or biochemical processes. The major factors responsible for causing disease in animals and humans are listed also below. All of them affect one or more critical chemical reactions or molecules in the body. Numerous examples of the biochemical basis of diseases will be encountered in this text. Okay. Now, additional examples of many of these uses are presented in various sections of this text. Now, figure 1, that's one of the diagrams we want to be looking at, is examples of the two-way street connecting biochemistry and medicine. Now, we have upper side, we have biochemistry, down we have medicine. In the upper street, we have nucleic acid. The lower street, we have genetic disease. Upper street, proteins. Lower street, sickle cell anemia. Upper street, we have lipid. Lower street, we have atherosclerosis, right? Upper street, carbohydrate. Lower street, we have diabetes mellitus. So, as a heading, impact of the Human Genome Project on biochemistry and medicine. Remarkable progress was made in the late 1990s in sequencing the human genome. This culminated in July 2000 when leaders of the two groups involved in this effort, the International Human Genome Sequencing Consortium and Celera Genomics, a private company, announced that over 90% of the genome had been sequenced. Draft versions of the sequence were published. So, in summary, biochemistry is the science concerned with studying the various molecules that occur in living cells and organisms with their chemical reaction, because life depends on biochemical reactions. Biochemistry has become the basic language of all biologic references. Biochemistry is concerned with the entire spectrum of life, from relatively simple viruses and bacteria to complex human beings. Biochemistry and medicine are intimately related. Health depends on a harmonious balance of biochemical reactions occurring in the body, and disease reflects abnormalities in biomolecules, biochemical reactions, or biochemical processes. Advances in biochemical knowledge have illuminated many areas of medicine. Conversely, the study of disease has often reviewed previous suspected aspects of biochemistry. The determination of the sequence of the human genome, nearly complete, will have a great impact on all areas of biology, including biochemistry, bioinformatics, and biotechnology. Biochemical approaches are often fundamental in illuminating the causes of diseases in designing appropriate therapies. The judicious use of biochemical laboratory tests is an integral component of diagnosis and monitoring of treatment. Its high knowledge of biochemistry and of other related basic disciplines is essential for the rational practice of medical and related health sciences. Now, in chapter 2, we have water and pH. This chapter is written by Victor W. Rodwell, PhD, and Peter J. Kennelly, PhD. Biomedical importance. Water is a predominant chemical component of living organisms. Its unique physical properties, which include the ability to solvate a wide range of organic and inorganic molecules, derived from water's dipolar structure and exceptional capacity for forming hydrogen bonds. The manner in which water interacts with a solvated biomolecule influences the structure of each. An excellent nucleophile, water is a reactant or product in many metabolic reactions. Water has a slight propensity to dissociate into hydroxide ions and protons. The acidity of aqueous solution is generally reported using the logarithmic pH scale. Bicarbonate and other buffers normally maintain the pH of extracellular field between 7.35 and 7.45. Suspected disturbances of acid-base balance are verified by measuring the pH of arterial blood and the CO2 content of venous blood. Causes of acidosis, that's when blood pH is less than 7.35, include diabetic ketosis and lactic acidosis. Alkalosis, pH greater than 7.5, may, for example, follow vomiting of acidic gastric content. Regulation of water balance depends upon hypothalamic mechanism that controls test on antidiuretic hormone, or retention or excretion of water by kidney, and on evaporative loss. Nephrogenic diabetes insipidus, which involves the inability to concentrate urine or adjust to subtle changes in extracellular fluid osmolarity, results from the unresponsiveness of renal tubular osmoreceptors to ADH. As a heading, water is an ideal biologic solvent. Water molecules form dipoles. A water molecule is an irregular, slightly skewed tetrahedron with oxygen as its center. The two hydrogen and the unshared electrons of the remaining two sp3 hybridized orbitals occupy the corners of the tetrahedron. The 105-degree angle between the hydrogen differs slightly from the ideal tetrahedral angle, 109.5 degree. Ammonia is also tetrahedral with a 107-degree angle between its hydrogens. Water is a dipole, a molecule with electric charge distribution asymmetrically about its structure. The strongly electronegative oxygen atom pulls electrons away from the hydrogen nuclei, leaving them with a partial positive charge, while its two unshared electron pairs constitute a region of local negative charge. Water is a strong dipole, has a high dielectric constant as described quantitatively by Coulomb's law. The strength of interaction F between oppositely charged particles is inversely proportionate to the dielectric constant epsilon of the surrounding medium. The dielectric constant for a vacuum is unity. For hexane it is 1.9, for ethanol it is 24.3, and for water it is 78.5. Water therefore greatly decreases the force of attraction between charged and polar species relative to water-free environment with lower dielectric constants. Its strong dipole and high electric constant enable water to dissolve large quantities of charged compounds such as salts. As a heading, water molecules form hydrogen bond. An unshielded hydrogen nucleus covalently bound to an electron withdrawing oxygen or nitrogen atom can interact with an unshared electron pair on another oxygen or nitrogen atom to form hydrogen bond. Since water molecules contain both of these features, hydrogen bonding favors the self-association of water molecules into ordered arrays. Hydrogen bonding profoundly influences the physical properties of water and accounts for its exceptionally high viscosity, surface tension, and boiling point. On average, each molecule in liquid water associates through hydrogen bond with 3.5 others. These bonds are both relatively weak and transient. With a half-life of about 1 microsecond, rupture of a hydrogen bond in liquid water requires only about 4.5 kcal per mole, less than 5% of the energy required to rupture covalent OH bond. Hydrogen bonding enables water to dissolve many organic biomolecules that contain functional groups which can participate in hydrogen bonding. The oxygen atoms of aldehydes, ketones, and amides provide pairs of electrons that can serve as hydrogen acceptors. Alcohols and amines can serve both as hydrogen acceptors and as donors of unshaded hydrogen atoms for formation of hydrogen bonds. If you don't have this book, or if you have it and you want to join me, this is Harper's Illustrated Biochemistry, 26th edition. 26th edition. I don't know. 26th edition. So That is that. So let's continue. I'm in page 6 now. Now, see heading, interaction with water influences the structure of biomolecules. Now, covalent and non-covalent bonds stabilizes biologic molecules. The covalent bond is the strongest force that holds molecules together. Non-covalent force, while of lesser magnitude, makes significant contributions to the structure, stability, and functional competence of macromolecules in living cells. These forces, which can be either attractive or repulsive, involve interaction both within the biomolecules and between it and the water that forms the principal component of the surrounding environment. As a heading, biomolecules fold to position polar and charged groups on their surfaces. Most biomolecules are amphipathic, that is, they possess regions rich in charged or polar functional groups, as well as regions with hydrophobic character. Proteins tend to fold with the R group of amino acids hydrophobic side chains in the interior. Amino acids with charged or polar amino acid side chains, example arginine, glutamate, serine, generally are present on the surface in contact with water. A similar pattern prevails in the phospholipid bilayer, where the charged head groups of phospho, phosphatidyl, oh, serine or phosphatidyl ethanolamine contact water, while their hydrophobic fatty acid side chains cluster together. I'm sorry for those mistakes, excluding water. This pattern maximizes the opportunities for the formation of energetically favorable charged dipole, and hydrogen bonding interactions between polar groups on the biomolecule and water. It also minimizes energetically unfavorable contact between water and hydrophobic groups. Okay, let's look at the hydrophobic interactions. Hydrophobic interaction refers to the tendency of nonpolar compounds to self-associate in an aqueous environment. This self-association is driven neither by mutual attraction nor by what are sometimes incorrectly referred to as hydrophobic bonds. Self-association arises from the need to minimize energetically unfavorable interaction between nonpolar group and water. While the hydrogen of nonpolar groups, such as the methylene groups of hydrocarbons, do not form hydrogen bonds, they do affect the structure of the water that surround them. Water molecules adjacent to a hydrophobic group are restricted in the number of orientations, that's degree of freedom, that permit them to participate in the maximum number of energetically favorable hydrogen bonds. Maximal formation of multiple hydrogen bonds can be maintained only by increasing the order of adjacent water molecules with a corresponding decrease in entropy. It follows from the second law of thermodynamics that optimal free energy of a hydrocarbon-water mixture is a function of both maximum enthalpy and minimum entropy. Thus, nonpolar molecules tend to form droplets with minimal exposed surface area, reducing the number of water molecules affected. For the same reason, in the aqueous environment of the living cell, the hydrophobic portions of biopolymers tend to be buried inside the structure of the molecule or within a lipid bilayer, minimizing contact with water. Electrostatic interaction as a heading. Interaction between charged groups shaped by molecular structure. Electrostatic interaction between oppositely charged groups within or between biomolecules are termed subbridges. Subbridges are compatible in the strength to hydrogen bond but act over larger distances. They thus often facilitate the binding of charged molecules and ions to proteins and nucleic acids. As a heading, van der Waals forces. Now, van der Waals forces arise from attraction between transient dipole united by rapid movement of electrons on all neutral atoms, significantly weaker than hydrogen bonds but potentially extremely numerous. Van der Waals forces decreases as they cease power of the distance separating atoms. Thus, they act over very short distances, typically 2 to 4 Armstrong. Multiple forces stabilize biomolecules as a heading. The DNA double helix illustrates the contribution of multiple forces to the structure of biomolecules. While each individual DNA strand is held together by covalent bond, the two strands of the helix are held together exclusively by non-covalent interaction. These non-covalent interactions include hydrogen bonds between nucleotide bases, that's Wasson-Crick base pairing, and van der Waals forces interaction between the stacked purine and pyrimidine bases. The helix presents the charged phosphate groups and polar ribose sugars of the backbone to water while bearing the relatively hydrophobic nucleotide base inside. The extended backbone maximizes the distance between negatively charged backbone phosphates, minimizing unfavorable electrostatic interaction. As a heading, water is an excellent nucleophile. Metabolic reactions often involve the attack by lone pairs of electron on electron-rich molecules termed nucleophiles on electron-poor atoms called electrophiles. Nucleophiles and electrophiles do not necessarily possess a formal negative or positive charge. Water, those two lone pairs of sp3 electrons bear a partial negative charge, is an excellent nucleophile. Other nucleophiles of biologic importance include the oxygen atoms of phosphates, alcohols, and carboxylic acid, the sulfur of thiols, the nitrogen of amines, and the imidazole ring of histidine. Common electrophiles include the carbonic carboninamides, esters, aldehydes, and ketones, and the phosphorus atoms of phosphoesters. Nucleophilic attack by water generally results in the cleavage of the amide, glycoside, or ester bond that hold biopolymers together. This process is termed hydrolysis. Conversely, when monomer units are joined together to form biopolymers such as proteins or glycogen, water is a product. While hydrolysis is a thermodynamically favored reaction, the amide and phosphoester bonds of polypeptides and oligonucleotides are stable in the aqueous environment of the cell. The seemingly paradoxical behavior reflects the fact that the thermodynamic governing the equilibrium of a reaction do not determine the rate at which it will take place. In the cell, protein catalysts called enzymes are used to accelerate the rate of hydrolytic reactions when needed. Proteases catalyze the hydrolysis of proteins into their component amino acids, while nucleases catalyze the hydrolysis of the phosphoester bonds in DNA and RNA. Careful control of the activities of this enzyme is required to ensure that they act on appropriate target molecules. Many metabolic reactions involve group transfer as a heading. In group transfer reaction, a group G is transferred from a donor D to an acceptor A, forming an acceptor group complex AG. The hydrolysis and phosphorylasis of glycogen represent group transfer reactions in which glycosyl groups are transferred to water or to orthophosphate. The equilibrium constant for the hydrolysis of covalent bonds strongly favors the formation of split products. The biosynthesis of macromolecules also involve group transfer reaction in which the thermodynamically unfavored synthesis of covalent bond is coupled to favored reaction, so that the overall change in free energy favors biopolymer synthesis. Given the nucleophilic character of water and its high concentration in cells, why are biopolymers such as protein and DNA relatively stable, and how can synthesis of biopolymers occur in apparently aqueous environments? Now, central to both questions are the properties of enzymes. In the absence of enzyme catalysis, even the thermodynamically high favored reactions do not necessarily take place rapidly. Precise and differential control of enzyme activity and the sequestration of enzymes in specific organelles determine under what physiologic condition a given biopolymer will be synthesized or degraded. Newly synthesized biopolymers are not immediately hydrolyzed, in part because the active sites of biosynthetic enzymes are sequestered substrates in an environment from which water can be excluded. As a heading, water molecules exhibit a slight but important tendency to disassociate. The ability of water to ionize, while slight, is of central importance for life. Since water can act as both as an acid and as a base, its ionization may be represented as intermolecular proton transfer that forms a hydronium ion and a hydroxide ion. The transferred protein is actually associated with a cluster of water molecules. Proton exist in solution not only as H3O+, but also as multimers such as H5O2+ and H7O3+. The proton is nevertheless routinely represented as H+, even though it is in fact highly hydrated. Since hydronium and hydroxide ions continuously recombine to form water molecules, an individual hydrogen and oxygen cannot be stated to be present as an ion or as a part of water molecule. At one constant, it is an ion. An instant later, it is part of the molecule. Individual ions or molecules are therefore not considered. We refer instead to the probability that in any instant, the time a hydrogen will be present as an ion as a part of a water molecule. Since 1 gram of water contains 3.46 times 10 raised to the power 22 molecules, the ionization of water can be described statistically. To state that the probability that a hydrogen exists as an ion is 0.01 means that a hydrogen atom has 1 chance in 100 of being an ion and 99 chances out of 100 of being part of water molecule. The actual probability of a hydrogen atom in pure water existing as a hydrogen ion is approximately 1.8 times 10 raised to the power minus 9. The probability of it being part of a molecule thus is almost unity. Stated another way, for every hydrogen ion and hydrogen ion in pure water, there is 1.8 billion or 1.8 times 10 raised to power 9 water molecules. Hydrogen ion and hydrogen ion nevertheless contribute significantly to the properties of water. For the dissociation of water, we have a particular reaction here where we have constant equals concentration of hydrogen ion, concentration of hydrogen ion all over concentration of water. And they calculated something, get the constant of water, which is 1.00 times 10 raised to the power minus 14 mole per liter square. And then continue. So pH is the negative log of the hydrogen ion concentration. This is how pH was calculated. That we have pH equal to negative log of concentration of hydrogen ion, right? So that is that. So now we want to look at functional groups that are weak acids have great physiologic significance. Many biochemicals possess functional groups that are weak acids or bases. Carboxyl groups, amino groups, and the second phosphate dissociation of phosphate esters are present in proteins and nucleic acid. Most enzymes and most intermediary metabolites. Knowledge of the dissociation of weak acid and the base thus is basic to understanding the influence of intracellular pH on structure and biologic activity. Charge-based separations such as electrophoresis and ion exchange chromatography also are best understood in terms of the dissociation behavior of functional groups. We term the protonated species the acid and the unprotonated species its conjugate base. Similarly, we may refer to a base and its conjugate acid. Representative weak acids, their conjugate base and the pKa values include the following. They wrote it down here. I'm reading, so there's no way I can read the equations out. So let's go to Henderson-Hesselbach equation. Describes the behavior of weak acid and buffers. That's the heading. Now, the Henderson-Hesselbach equation is derived below. A weak acid H ionizes as follows. There we have H equals H plus A minus the equilibrium constant. This is also the derivation of the Henderson-Hesselbach equation, where we have at the end of the day, we have a particular equation where we have pH equals pKa plus log concentration of A minus all over concentration of HA. Right? Good. So now solutions of weak acid and their salts buffer changes to pH as a heading. Solution of weak acids or bases and their conjugate exhibit buffering. The ability to resist a change in pH following addition of strong acid or strong base. Since many metabolic reactions are accompanied by the release or uptake of protons, most intracellular reactions are buffered. Oxidative metabolism produces CO2, the anhydride of carbonic acid, which if not buffered would produce severe acidosis. Maintenance of a constant pH involves buffering by phosphates, bicarbonate, and proteins, which accept or release protons to resist a change in pH. For experiments using tissue extracts or enzymes, constant pH is maintained by the addition of buffers such as MES, that's 2N-morpholino, ethansulfonic acid, where pKa is 6.1, inorganic orthophosphate, pKa 7.2, HEPES, that is N-hydroxyethylpiperazine, N2-ethansulfonic acid, pKa 6.8, or TRIS, that's 3-hydroxymethylamino-methane, pKa 8.3. The value of pKa relative to the desired pH is the major determinant of which buffer is selected. Buffering can be observed by using a pH meter while titrating a weak acid or base. We can also calculate the pH shift that accompanies addition of acid or base to a buffer solution. So, now I said, in the example, the buffered solution, a weak acid pK equals 5.0 and its conjugate base is initially at one of 4 pH values. We will calculate the pH shift that results when 0.1 of KOH is added to 1 of each solution. This is also mathematical equation. This is biochemistry, right? So now acid strength depends on molecular structure. Many acids of biologic interest possess more than one dissociating group. The presence of adjacent negative charge hinders the release of a proton from a nearby group, rising to its pKa. This is apparent from the pKa values from the three dissociating groups of phosphoric acid and citric acid. The effect of adjacent charge decreases with distance. The second pKa for succinic acid has two methylene groups between its carboxyl group is 5.6, where the second pKa for glutaric acid, which has one additional methylene group, is 5.4. Gone 30 minutes. Let's continue. pKa values depend on the properties of the medium. As a heading, the pKa of a functional group is also profoundly influenced by the surrounding medium. The medium may either rise or lower the pKa depending on whether the undissociated acid or its conjugate base is the charged species. The effect of the electric constant on pKa may be observed adding ethanol to water. The pKa of a carboxylic acid increases, whereas that of an amine decreases, because ethanol decreases the ability of water to solvate a charged species. The pKa values of dissociating groups in the interiors of protein thus are profoundly affected by their local environment, including the presence or absence of water. Now, in summary, water forms hydrogen bonded clusters where it serves with other proton donors or acceptors. Hydrogen bonds account for the surface tension, viscosity, liquid state at room temperature, and solvent power of water. Compounds that contain ON or S can serve as hydrogen bond donors or acceptors. Macromolecule exchange internal surface, hydrogen bonds for hydrogen. Let's go again. Macromolecule exchange internal surface hydrogen bonds for hydrogen bonds to water. Entropic forces dictate that macromolecules expose polar regions to an aqueous interface and bury non-polar regions. Short bonds, hydrophobic interaction, and van der Waals forces participate in maintaining molecular structure. pH, the negative logarithm of hydrogen ion. A lower pH characterizes an acidic solution, and a high pH denotes a basic solution. The strength of weak acid is exposed by pKa, the negative logarithm of the acid dissociation constant. Strong acid have low pKa values and weak acid have high pKa values. Buffer resist a change in pH when protons are produced or consumed. Maximum buffering capacity occurs in plus or minus 1 pH unit on either side of pKa. Physiologic buffers include bicarbonate, orthophosphate, and proteins. So we're done with the second chapter. Let's go to the third chapter, amino acids and peptides. Now, this chapter was written by Victor W. Rodwell, PhD, and Peter J. Kennelly, the same people that wrote the chapter 2. So, biomedical importance. In addition to providing the monomer units from which the long polypeptide chains of protein are synthesized, the alpha amino acids and the derivatives participate in cellular functions as diverse as nerve transmissions and the synthesis of porphyrins, purines, pyrimidines, and urea. Short polymers of amino acids called polypeptides perform prominent roles in the neuroendocrine systems as hormones, hormone-releasing factors, neuromodulators, or neurotransmitters. While proteins contain only L-alpha amino acids, microorganisms elaborate peptides that contain both D and L-alpha amino acids. Several of these peptides are of therapeutic value, including the antibiotics bacitracin and gramicidin, E, and the antitumor agent bleomycin. Certain other microbial peptides are toxic. The cyanobacterial peptide, microcystin, and nodularin are lethal in large doses, while some quantities promote the formation of hepatic tumors. Neither humans nor any other higher animals can synthesize 10 of the 20 common L-alpha amino acids in amounts adequate to support infant growth or to maintain health in adults. Consequently, the human diet must contain adequate quantities of these nutritionally essential amino acids. Now, as a heading, properties of amino acids. Now, the genetic code specifies 20 L-alpha amino acids. When this book was written, I guess 20, but recently we find out that we have extra, which is selenocysteine, which makes it the 21st. I guess as we advance, more will be discovered too. So of over the 300 naturally occurring amino acids, 20 constitute the monomer unit of protein, while a non-redundant 3-letter generic code could accommodate more than 20 amino acids. This redundancy limits the available codons to the 20 alpha amino acids listed below. Both 1 and 3-letter abbreviations for each amino acid can be used to represent the amino acids in peptide. Some proteins contain additional amino acids that arise by modification of an amino acid already present in a peptide. Examples include conversion of peptidylproline and lysine to 4-hydroxyproline and 5-hydroxylysine, the conversion of peptidylglutamate to beta-carboxyglutamate, and the methylation, formation, acetylation, and prenylation, and phosphorylation of certain amino acid residues. This modification extends the biologic diversity of proteins by altering their solubility, stability, and interaction with other proteins. As a heading, only L-alpha amino acids occur in protein. With the sole exception of glycine, the alpha carbon of amino acid is chiral. Although some protein amino acids are dextrorotatory and some levorotatory, all share the absolute configuration of L-glyceraldehyde and thus are L-alpha amino acids. Several free L-alpha amino acids fulfill important roles in metabolic processes. Examples include ornithine, citrulline, and argininosuccinate that participate in urea synthesis, tyrosine in formation of thyroid hormones, and glutamate in neurotransmitter biosynthesis. The amino acids that occur naturally include free-glycerin and D-aspartate in brain tissue, D-alanine and D-glutamate in the cell walls of gram-positive bacteria, and D-amino acid in some non-mammalian peptides and certain antibiotics. And let's look at the different amino acids as was written here. We have glycine, alanine, valine, leucine, isoleucine. The amino acid with aliphatic acid side chains. Now, with side chains containing hydroxyl groups, we have serine, pyrroline, tyrosine. With side chains containing sulfur atoms, we have cysteine or cysteine, methionine. With side chains containing acidic groups or their amines, we have aspartic acid, asparagine, glutamic acid, glutamine. Now, with side chains containing basic groups, we have arginine, lysine, histidine. With side chains containing with side chains containing aromatic rings, we have histidine, phenylalanine, tyrosine, tryptophan. With side chains containing amino acid, we have proline. Now, as a heading, amino acid may have positive, negative, or zero net charged. Charged and uncharged forms of the ionizable COOH and NH3 plus weak acid groups exist in solution in protonic equilibrium. Now, molecules that contain an equal number of ionizable groups of opposite charge and that therefore bear no net charge are termed zwittrions or wittrions if the Z is silent. Amino acid in blood and most tissue are represented below. So there is a particular structure here, complex structure. I can't explain structures now. So it's in page 17 of the book. If you have this book, you see it there. So pKa value expresses the strength of weak acids. That's the heading. The acid strength of weak acids are expressed as their pKa value. The imidazole group of histidine and the guanidine group of arginine exist as resonance hybrids with positive charge distributed between both nitrogens, that's histidine, on all 3 nitrogens, arginine. The net charge of an amino acid, the algebraic sum of all positively and negatively charged groups present, depends upon the pKa values of its functional groups and on the pH of the surrounding medium. Altering the charge on amino acids and their derivatives by varying the pH facilities the physical separation of amino acids, peptides, and proteins. Now, at its isoelectric pH, that's pi, an amino acid bears no net charge. Let's discuss that. Let's read it. I hope you can hear me. This is not easy. Now, the isoelectric species is the form of a molecule that has an equal number of positive and negative charges and thus is electrically neutral. The isoelectric pH, also called the pi, is the pH midway between pKa values on the either side of the isoelectric species For an amino acid such as alanine that has only two dissociating groups, there is no ambiguity. The first pKa is 2.35 and the second pKa is 9.69. Now they gave a particular equation of isoelectric pH of alanine. We have Pi, that's isoelectric pH, because pK1 plus pK2 over 2. And then they said that pK1 is 2.35, pK2 is 9.69 over 2. We have 6.02 and all that. So let's continue. pKa values vary with the environment. The environment of a dissociable group affects its pKa. The pKa values of the R groups of free amino acids in aqueous solution thus provide only an approximate guide to the pKa values of the same amino acid when present in proteins. A polar environment favors the charged form, that's RCOO- or RNH3+, and a non-polar environment favors the uncharged form, RCOOH or RNH2. A non-polar environment thus raises the pKa of the carboxyl group, making it a weaker acid, but lowers that of amino acids, making it stronger acid. The presence of adjacent charged groups can reinforce or counteract solvent effect. The pKa of a functional group thus will depend upon its location within given protein. Variations in pKa can encompass whole pH units. pKa values that diverge from those listed by as much as 3 pH units are common at the active sites of enzymes. An extreme example, a buried aspartic acid of thyrodoxine has a pKa above 9, a shift over 6 pH units. The solubility and melting point of amino acids reflects their ionic character. That's the heading. The charged functional groups of amino acids ensure that they are readily solvated by and thus soluble in polar solvents such as water and ethanol, but insoluble in non-polar solvents such as benzene, hexane, or ether. Similarly, the high amount of energy required to disrupt the ionic forces that stabilize the crystal lattice account for account for the high melting point of amino acids greater than 200 degrees C. Amino acids do not absorb visible light and thus are colorless. However, tyrosine, Ferdinand, and especially tryptophan absorb high wavelength 2050 to 2090 nanometer. Now, ultraviolet light. Tryptophan therefore makes the major contribution to the ability of most proteins to absorb light in the region of 280 nanometer. Now, as a heading, the alpha R groups determine the properties of amino acids. Since glycine, the smallest amino acids, can be accommodated in places inaccessible to other amino acids, it often occurs where peptides can bend sharply. The hydrophobic R groups of alanine, valine, leucine, and isoleucine, and the aromatic R groups of phenylalanine, tyrosine, and tryptophan typically occur primarily in the interior of cytosolic proteins. Cytosolic proteins. The charged R groups of basic and acidic amino acids stabilize specific proteins conformations via ionic interactions or salt bonds. These bonds also function in charged relay systems during enzymatic catalysis and electron transport in respiring mitochondria. Histidine plays a unique role in enzymatic catalysis. The pKa of its imidazole protein permits it to function at neutral pH as either a base or an acid catalyst. The primary alcohol group of serine and the primary thioalcohol, that's SH group of cysteine and excellent nucleophile, and can function as are excellent nucleophiles and can function as such during enzymatic catalysis. However, the secondary alcohol group of threonine, while a good nucleophile, does not fulfill an analogous role in catalysis. The OH groups of serine, tyrosine, and threonine also participate in regulation of the activities of enzymes, whose catalytic activity depends on the phosphorylation state of the residues. Okay, let's continue. Functional groups dictate the chemical reactions of amino acid. Each functional group of an amino acid exhibits all of its characteristics chemical reactions. For carboxylic acid groups, these reactions include the formation of esters, amines, and acid anhydrides. For amino acid acylation, amidation, and esterification, and for OH and SH groups, oxidation and what? Esterification. The most important reaction of amino acid is the formation of a peptide bond. As a heading, amino acid sequence determines primary structure. The number and order of all the amino acid residues in a polypeptide constitute its primary structure. Amino acid residue present in peptides are called amino acid residue and are named by representing the 8 or INE suffixes of free amino acid with YL, example alanine, aspartyl, tyrosine. Peptides are then named as derivatives of the carboxyl terminal amino acid residue. For example, lysyl-tyrgluone is called lysyl-leucine-tyrosylglutamine. The INE ending on glutamine indicates that the alpha carboxyl group is not involved in peptide bond formation. Peptide structures are easy to draw as a heading. Prefixes like tri or octa denote peptide with 3 or 8 residues, respectively, not those with 3 or 8 residue bonds, but by convention, peptides are written with the residue that bears the free alpha amino group at the left rather. To draw peptide, use a zigzag to represent the main chain or backbone. Add the same main chain atoms which occur in the repeating order: alpha nitrogen, alpha carbon, carbonyl carbon. Now add a hydrogen atom to each alpha carbon and to each peptide nitrogen and an oxygen to the carbonyl carbon. Finally, add the appropriate allyl groups to each alpha carbon. Now, three-letter abbreviations linked by straight line represent an unambiguous primary structure lines are omitted in single letter. This is just the diagram. Now, some peptide certain contain unusual amino acids. In mammals, peptide hormones typically contain only the alpha amino acids of proteins linked by standard peptide bonds. Other peptides may, however, contain non-protein amino acid derivatives of the protein amino acids or amino acid linked by an atypical protein bond. For example, the amino acid glutamate of glutathione, which participates in protein folding and the metabolism of xenobiotics, is linked to cysteine by a non-alpha peptide bond. The amino terminal glutamate of thyrotropin-releasing hormone is cyclized to pyroglutamic acid, and the carboxyl group of the carboxyl terminal prolyl residue is amidated. Peptide elaborated by fungi, bacteria, and lower animals can contain non-protein amino acids. The antibiotics thyroxidine and gramicidine S are cyclic polypeptides that contain D-phenylalanine and thine. The heptapeptide opioids, dermorphine and deltaphorin in the skin of South American tree frogs contain D-tyrosine and D-alanine. My pen fell. So peptides are polyelectrolytes. Peptides are polyelectrolytes, and the peptide bond is uncharged at any pH of physiologic interest. Formation of peptides from amino acid is therefore accompanied by a net loss of one positive and one negative charge for peptide bond formed. Peptides nevertheless are charged at physiologic pH owing to their carboxyl and amino terminal groups, and where present, their acidic or basic allo groups. As for amino acid, the net charge on a peptide depends on the pH of its environment and on the pK value of its dissociating groups. Now, the peptide bond has partial double bond character as a heading. Although peptides are written as if single bond linked to the alpha carboxyl and alpha nitrogen atom, this bond in fact exhibits partial double bond character. There thus is no freedom of rotation about the hydrogen bond that connects the carbonyl carbon and nitrogen of a peptide bond. Consequently, all four of the colored atoms are coplanar. The imposed semi-rigidity of the peptide bonds has important consequences for higher orders of proven structure. Okay, let's continue. Now, as a head, the non-covalent forces constrain peptide conformations. Constrain peptide conformations as a heading. Folding of a polypeptide probably occurs coincident with its biosynthesis. The physiologically active conformation reflects the amino acid sequence, steric hindrance, and non-covalent interaction. Example, hydrogen bonding hydrophobic interactions between residues. Common conformations include alpha helices and beta pleated sheets. As a heading, analysis of the amino acid content of biological materials. In order to determine the identity and quantity of each amino acid in a sample of biological material, it is first necessary to hydrolyze the peptide bonds that link the amino acid together by treatment with hot HCl. The resulting mixture of free amino acid is then treated with the cis-amino N-hydroxysuccinimidyl carbamate, which reacts with the alpha amino groups, forming fluorescent derivatives that are then separated and identified using high-pressure liquid chromatography. Ninhydrin, also widely used for detecting amino acid, forms a purple product with alpha amino acid and a yellow adduct with the amine groups of proline and hydroxychloride. Now, summary of this chapter 3. Both D amino acid and N and non-alpha amino acids are in net what am I saying? Summary. Both D amino acid and non-alpha amino acids occur in nature, but only L alpha amino acids are present in proteins. All amino acids possess at least two weakly acidic functional groups, RNH3 plus and RCOH. Many also possess additionally weakly acidic functional groups such as OH, SH, guanidino, or imidazole group. The pKa values of all functional groups of an amino acid dictate its net charge at a given pH. Pi is the pH at which amino acid bears no net charge and thus does not move in a direct current electric field. Of the biochemical reactions of amino acid, the most important is the formation of peptide bonds. The arrow groups of amino acid determine their unique biochemical functions. Amino acids are classified as basic, acidic, aromatic, aliphatic, or sulfur-containing based on the properties of the arrow groups. Peptides are named for the number of amino acid residue present and as derivatives of the carboxyl terminal residue. The primary structure of a peptide is its amino acid sequence, starting from the amino terminal residue. The partial double bond character of the bond that links the carbonyl carbon and nitrogen of a peptide renders 4 atoms of the peptide bond coplanar and restricts the number of possible peptide conformations. So this chapter 4, I didn't see where it started. My chapter 3 stopped at page 20. So chapter 4, I didn't see where it started, but this is page 25 on my face here. Let me show you. This is page 25. This is chapter, page 20. You can see it back. So I can't see the chapter 4 here. It's like it's torn out. So I want to continue with that page 25 since the chapter 4 is not here. This is chapter 4 too, but I'm talking about where it started. It's not there. So now separated the A and B chains are cleaved each chain into smaller peptides using trypsin, chymotrypsin, and pepsin. The resulting peptides were then isolated, treated with acid to hydrolyze peptide bonds and generalize peptides with as few as 2 or 3 amino acids. Each peptide was reacted with 1-fluoro-2,4-dinitrobenzene, Sanger's reagent, which derivatizes the exposed alpha amino groups of amino acid residues. The amino acid content of each peptide was then determined, while the beta amino acid of lysine also reacts with Sanger's reagent. Amino terminal lysine can be distinguished from those at other positions because they react with 2 mole of Sanger's reagent. Working backward to larger fragments enables Sanger to determine the complete sequence of insulin, an accomplishment for which he received a Nobel Prize in 1958. As a heading, the Edman reaction enables peptides and proteins to be sequenced. Per Edman introduced phenylisothiocyanate, Edman's reagent, Edman's reagent, to selectively label the amino terminal residue of a peptide. In contrast to Sanger's reagent, the phenylthiohydantoin (PTH) derivative can be removed under mild conditions to generate a new amino terminal residue. Successive rounds of derivatives with Edman's reagents can therefore be used to sequence many residues of a single sample of peptide. Edman sequencing has been automated using a thin film or solid matrix to immobilize the peptide, and HPLC to identify PTH amino acid. Modern gas sequencers can analyze as little as a few picomole of peptides. As a heading, large polypeptides are first clipped into smaller segments. While the first 20 to 30 residues of a polypeptide can readily be determined by the Edman method, most polypeptides contain several hundred of amino acids. Consequently, most polypeptides must first be cleaved into smaller peptides prior to Edman sequencing. Cleavage also may be necessary to circumvent post-translational modification that render a protein's alpha amino group blocked or unreactive with the Edman's reagent. It usually is necessary to generate several peptides, usually more than one method of cleavage. This reflects both inconsistency in the spacing of chemically or enzymatically susceptible cleavage sites and the need for sets of sequence of the polypeptide from which they derive. Reagents for the chemical or enzymatic cleavage of protein include cyanogen bromide (CNBr), trypsin, and Staphylococcus aureus (V8 protease). Following cleavage, the resulting peptides are purified by reverse phase (HPLC) or occasionally by SDS-PAGE and sequenced. As a heading, molecular biology has revolutionized the determination of primary structure. Knowledge of DNA sequences permits the deduction of the primary structure of polypeptides. DNA sequencing requires only minute amounts of DNA and can readily yield the sequence of hundreds of nucleotides. To clone and sequence the DNA that encodes a particular protein, some means of identifying the correct clone, example, knowledge of a protein proportion of which nucleotide sequence is essential. A hybrid approach thus has emerged. Edman sequencing is used to provide a partial amino acid sequence. Oligonucleotide primers modeled on the partial sequence can then be used to identify clones or to amplify the appropriate gene by the polymerase chain reaction (PCR). Once an authentic DNA clone is obtained, its oligonucleotide sequence can be determined and the genetic code used to infer the primary structure of the encoding polypeptide. The hybrid approach enhances the speed and efficiency of primary structure analysis and the range of proteins that can be sequenced. It also circumvents obstacles such as the presence of an amino terminal blocking group or the lack of key overlap peptides. Only a few segments of primary structure must be determined by Edman analysis. DNA sequencing reveals the order in which amino acids are added to the nascent polypeptide chain as it is synthesized on the ribosomes. However, it provides no information about post-translational modification such as proteolytic processing, methylation, glycosylation, phosphorylation, hydrolysation of proline and lysine, and disulfide bond formation that accompany maturation. While Edman sequencing can detect the presence of most post-translational events, technical limitations often prevent identification of spatial modification. Now, as a heading, mass spectrometry dictates covalent modification. Mass spectrometry detects covalent modification. Mass spectrometry, which discriminates molecules based solely on their mass, is ideal for detecting the phosphate, hydroxy, and other groups on post-translational modified amino acids. Each has a specific and readily identified increment of mass to the modified amino acid. For analysis by mass spectrometry, a sample in a vacuum is vaporized under conditions where protonation can occur, imparting positive charge. An electric field then propels the cation through a magnetic field which deflects them at a right angle to their original direction of flight and focuses them onto a detector. The magnetic force required to deflect the path of each ionic species onto a detector measured as the current applied to the electromagnet is recorded. For ions of identical net charge, this force is proportionate to their mass. In a time of flight, mass spectrometer, a briefly applied electric field accelerates the ion towards a detector that records the time at which each ion arrives. For molecules of identical charge, the velocity to which they are accelerated and hence the time required to reach the detector will be inversely proportionate to their mass. Conventional mass spectrometers generally are used to detect the masses of molecules of 1000 Dalton or less, whereas time-of-flight mass spectrometers are suited for determining the large masses of proteins. The analysis of peptides and proteins by mass spectrometry initially was hindered by difficulties in volatilizing large organic molecules. However, matrix-assisted laser desorption and electrospray dispersion permit the masses of even large polypeptides to be determined with extraordinary accuracy. Using electrospray dispersion, peptides eluting from a reverse phase HPLC column are introduced directly into the mass spectrometer for intermediate determination. Okay. Peptides inside the mass spectrometer are broken down into smaller units by collision with neutral helium atoms, and the masses of the individual fragments are determined. Since peptide bonds are much more labile than carbon-carbon bonds, the most abundant fragments will differ from one amino acid by units, equivalent to one or two amino acids. Since, with the exception of leucine and isoleucine, the molecular mass of each amino acid is unique, the sequence of the polypeptide can be reconstructed from the mass of its fragments. So we are 1 hour plus in this challenge. I'm happy. Let's keep going. So tandem mass spectroscopy. Complex peptide mixture can now be analyzed without prior purification by tandem mass spectrometry. Linked in series, spectrometers linked in series. The first spectrometer separates individual peptides based upon their differences in mass by adjusting the field strength of the first. Okay. By adjusting, by adjusting the field strength of the first magnet, a single peptide can be directed into the second mass spectrometer, where fragments are generated and their mass is determined. As the sensitivity and versatility of mass spectrometry continues to increase, it is displacing Edman sequencers for the direct analysis of protein primary structure. Now, as a heading, genomics enables proteins to be identified from a small amount of sequence data. Primary structure analysis has been revolutionized by genomics. The application of automated oligonucleotide sequencing and computerized data retrieval and analysis to sequence an organism's entire genetic complement. The first genome sequenced was that of Haemophilus influenza in 1995. By mid-2001, the complete genome sequences for over 50 organisms had been determined. These include the human genome and those of several bacterial pathogens. The results and significance of the Human Genome Project are discussed in the later chapters. Where genome sequence is known, the task of determining a protein's DNA derived primarily sequence is materially simplified. In essence, the second half of the hybrid approach has already been completed. All that remains is to acquire sufficient information to permit the open reading frame that encodes the protein to be retrieved from an internet accessible genome database and identified. In some cases, a segment of amino acid sequence only 4 or 5 residues in length may be sufficient to identify the correct ORF. Computerized search algorithms assist the identification of the gene encoding a given protein and clarifying uncertainties that arise from Edman's sequencing and mass spectrometry. By exploiting computers to solve complex puzzles, the spectrum of information suitable for identification of the ORF that encodes a particular polypeptide is greatly expanded. In peptide mass profiling, for example, a peptide digest is introduced into the mass spectrometer and the size of the peptides are determined. A computer is then used to find an ORF whose predicted protein product would, if broken down into peptides by the cleavage method selected, produce a set of peptides whose masses match those observed mass spectrometry. Proteomics and the proteome as a heading. Now, the goal of proteomics is to identify the entire complement of proteins elaborated by a cell under diverse conditions. While the sequence of the human genome is known, the picture provided by genomics alone is both static and incomplete. Proteomics aim to identify the entire complement of proteins elaborated by a cell under diverse conditions. A gene are switched on and off Proteins are synthesized in particular cell types at specific times of growth or differentiation and in response to external stimuli. Muscle cells express proteins not expressed by neutral cells, and the type of subunits present in the hemoglobin tetramer undergo change pre and postpartum. Many proteins undergo post-translational modification during maturation into functionally competent form or as a means of regulating their properties. Knowledge of the human genome therefore represents only the beginning of the task of describing living organisms in molecular detail and understanding the dynamics of processes such as growth, aging, and diseases. As the human body contains thousands of cell types, each containing thousands of proteins, the proteome, the set of all the proteins expressed by an individual cell at a particular time, represents a moving target of formidable dimensions. As a heading, two-dimensional electrophoresis and gene array chips are used to survey protein expressions. One goal of proteomics is the identification of the proteins whose level of expression correlates with medically significant events. The presumption is that proteins whose appearance or disappearance is associated with a specific physiologic condition or disease will provide insight into root causes and mechanisms. Determination of the proteome's characteristics of each cell type requires the utmost efficiency in the isolation and identification of individual proteins. The contemporary approach utilizes robotic automation to speed sample preparation on large two-dimensional gels to resolve cellular proteins. Individual polypeptides are then extracted and analyzed by Edman sequencing or mass spectroscopy. While only about 1,000 proteins can be resolved on a single gel, two-dimensional electrophoresis has a major advantage in that it examines the proteins themselves. An alternative and contemporary and complementary approach employs gene arrays, sometimes called DNA chips, to detect the expression of the mRNAs, which encodes protein. When changes in the expression of the mRNA encoding a protein do not necessarily reflect comparable changes in the level of the corresponding protein, gene arrays are more sensitive probes than two-dimensional gels and thus can examine more gene products. Bioinformatics assist identification of protein function. That's the heading. The function of a large proportion of the proteins encoded by the human genome are presently unknown. Recent advances in bioinformatics present researchers to compare amino acid sequences to discover clues to potential properties, physiologic roles and mechanism of action of proteins. Algorithms exploit the tendency of nature to employ variations of a structural team to perform similar functions in several proteins. Example, the Rossmann nucleotide binding fold to bind NADPH, nuclear targeting sequencing, and EF hands to bind Ca+. These domains generally are detected in the primary structure by observation of particular amino acids at key positions. Insight into the properties and physiologic role of a newly discovered protein may be thus may be inferred by comparing its primary structure with that of known protein. And for summary of chapter 4, long amino acid polymers or polypeptide constitute the basic structural unit of protein, and the structure of a protein provides insight into how it fulfills its function. The Edman reaction enables amino acid sequence analysis to be automated. Mass spectrometry provides a sensitive and versatile tool for determining primary structure and for identification of post-translational modification. DNA cloning and molecular biology coupled with protein chemistry provide a hybrid approach that greatly decreases the speed and efficiency for determination of primary structure of proteins. Now, genomics, the analysis of the entire oligonucleotide sequence of an organism's complete genetic material, has provided further enhancement. Computer algorithms facilitate the identification of open reading frames and encode a given protein by using a partial sequences and peptide mass profiling to search sequence database. Scientists are now trying to determine the primary sequence and functional role of every protein expressed in the living cell, known as its proteome. A major goal is the identification of proteins whose appearance or disappearance correlates with physiologic phenomena, aging, or specific diseases. Now, let's go to chapter 5. We just finished chapter 4. Yeah. Now, chapter 4, proteins, higher order of structure, higher orders of structures. Victor W. Rodwell, PhD, and Peter J. Kennedy, same people. Now, biomedical importance. Biomedical importance. Protein catalyze metabolic reactions, power cellular motion, and form micromolecules, rods, and cables that provide structural integrity to hair, bones, tendons, and teeth. In nature, form follows functions. The structural variety of human proteins therefore reflect the sophistication and diversity of their biologic roles. Maturation of a newly synthesized polypeptide into a biological functional protein requires that it be folded in a specific three-dimensional arrangement or conformation. During maturation, during maturation, post-translational modifications may add new chemical groups or remove transiently needed peptide segments. Genetic or nutritional deficiencies that impede protein maturation are deleterious to health. Examples of the former include Creutzfeldt-Jakob disease, scrapie, Alzheimer's disease, and bovine spongiform encephalopathy, mad cow disease. Scurvy represents a nutritional deficiency that impairs protein maturation. Now, conformation versus configuration. The term configuration and conformation are often confused. Configuration refers to the geometric relationship between a given set of atoms, for example, those that distinguish the L from the D amino acids. Interconversion of the configuration alternatives require breaking covalent bonds. Meanwhile, conformation refers to the spatial relationship of every atom in a molecule. Interconversion between conformers occur without covalent bond rupture, with retention of configuration and typically via rotation about single bonds. As a heading, proteins were initially classified by their gross characteristics. Scientists initially approached structure-function relationships in protein by separating them into classes based upon properties such as solubility, shape, or the presence of non-protein groups. For example, a protein that can be extracted from cells using solution at physiologic pH and ionic strength are classified as soluble. Extraction of integral membrane proteins require dissolution of the membrane with detergents. Global proteins are compact, are roughly spherical or ovoid in shape, and have axial ratios. The ratio of their shortest to longest dimension of not over 3. Most enzymes are globular proteins whose large internal volume provides ample space in which to construct cavities of the specific shape, charge, and hydrophobicity or hydrophilicity required to bind substrates and promote catalysis. By contrast, many structural proteins adopt highly extended conformations. These fibrous proteins possess axial ratios of 10 or more. Lipoproteins and glycoproteins contain covalently bound lipid and carbohydrates, respectively. Myoglobin, hemoglobin, cytochromes, and many other proteins contain tightly associated metal ions and are termed metalloprotein. With the development and application of techniques for determining the amino acid sequences of proteins, more precise classification schemes have emerged and based upon similar or a classification terms remain in common use. Now, as a heading, are constructed using modular principles. Proteins perform complex physical and catalytic function by positioning specific chemical groups in a precise three-dimensional arrangement. The polypeptide scaffold containing these groups must adopt a conformation that is both functionally efficient and physiologically strong. At first glance, the biosynthesis of polypeptide comprised of tens of thousands of individual atoms would appear to be extremely challenging. When one considers that a typical polypeptide can adopt greater than or equal to 10 raised to the power of 50 distinct conformations, folding into the conformation appropriate to their biologic function would appear to be even more difficult. As described in the last chapters 3 and 4, synthesis of the polypeptide backbones of protein employ a small set of common building blocks or modules, the amino acids, joined by a common linkage, the peptide bond. The stepwise modular pathway simplifies the folding and the processing of newly synthesized polypeptides into mature proteins. Now, as a heading, we have the four orders of the protein structure. The four orders of the protein structure. Now, a modular nature of protein synthesis and folding are embodying the concept of orders of protein structure. Primary structure, the sequence of the amino acid in polypeptide chain. Secondary structure, the folding of short 3 to 30 residues, contiguous segments of polypeptide into three geometrically ordered units. Tertiary structure, the three-dimensional assembly of secondary structural units to form larger functional units, such as the mature polypeptide and its component domains. And the quaternary structure, that's the number and the types of polypeptide units of oligomeric proteins and their special arrangements. Now, as a heading, secondary structure. Peptide bond restricts possible secondary conformations. Free rotation is possible about only 2 or 3 covalent bonds of the polypeptide backbone. The alpha carbon to the carbonyl carbon bond and the alpha carbon to nitrogen bond. The partial double bond character of the peptide bond that links carbonyl carbon to the alpha nitrogen requires that the carbonyl carbon, carbonyl oxygen, and alpha nitrogen remains coplanar. That prevents the rotation. That is, the angle between the alpha carbon and nitrogen bond is termed the phi bond or the phi angle, right? PHI, phi angle. And that about the carbonyl carbon and the alpha carbon bond the psi angle, PSI. For amino acids other than glycine, most combination of phi and psi angles are disallowed because of the steric hindrance, right? Because of the steric hindrance. The conformation of proline are even more restricted due to the absence of free rotation of the N alpha carbon bond. Regions of ordered secondary structure arise when a series of amino acid residues adopt similar phi and psi angles. Extended segments of polypeptide can possess a variety of such angles. The angles that define the two most common types of secondary structure, the alpha helix and the beta sheet, fall within the lower and upper left-hand quadrants of all Ramachandran plots, respectively. Now, as a heading, the alpha helix. A polypeptide backbone of an alpha helix is twisted by an equal amount about each alpha carbon with a phi angle of approximately 57 degrees and a psi angle of approximately 47 degrees. A complete turn of the helix contains an average of 3.6 amino acid residue and distance it rises per turn. Its pitch is 0.54 nanometer. The arrow groups of each amino acid residue in an alpha helix face outward. Proteins contain only L amino acid, for which a right-handed alpha helix is by far the most stable, and only right-handed alpha helices occur in nature. Schematic diagram of proteins represent alpha helix as cylinders. Now, the stability of alpha helix arises primarily from hydrogen bonds formed between the oxygen of the peptide bond carbonyl and the hydrogen atom of the peptide bond nitrogen of the fourth residue down the polypeptide chain. The ability to form the maximum number of hydrogen bonds supplemented by van der Waals interaction in the core of this tightly packed structure provides the thermodynamic driving force for the formation of an alpha helix. Since the peptide bond nitrogen of proline lacks a hydrogen atom to contribute to a hydrogen bond, proline can only be stably accommodated within the first turn of an alpha helix when present elsewhere. Proline disrupts the conformation of the helix, producing a bend, right? Because of its muscle, glycine also often induces bends in alpha helix. Many alpha helix have predominantly hydrophobic arrow groups on one side of the axis of the helix and predominantly hydrophilic ones on the other. These amphipathic helices are well adapted to the formation of an interfaces between polar and non-polar regions, such as the hydrophobic interior of a protein and its aqueous environment. Clusters of amphipathic helices can create a channel or pore that permit a specific polar molecules to pass through hydrophobic cell membranes. The beta sheet as a heading. The second, hence beta, recognizable regular secondary structure in protein is the beta sheet. The amino acid residue of a beta sheet, when viewed edge-on, form a zigzag or pleated pattern in which the arrow groups of adjacent residue point in opposite direction, unlike the compact backbone of the alpha helix. The peptide backbone of the beta sheet is highly extended, but like the alpha helix, beta sheets derive much of their stability from the hydrogen bonds between the carbonyl oxygens and the amide hydrogens of peptide bonds. However, in contrast to the alpha helix, these bonds are formed with adjacent segments of beta sheets. Interacting beta sheets can be arranged either to form a parallel beta sheet in which the adjacent segments of the polypeptide chain proceed in the same direction and minor to carboxyl, or an antiparallel sheet in which they proceed in opposite direction. Either configuration permits the maximum number of hydrogen bonds between segments of strand of the sheet. Most beta sheets are not perfectly flat but tend to have a right-handed twist. Clusters of twisted strands of beta sheets form the core of many globular proteins. Schematic diagram represents beta sheets as arrows that point in the amino acid carboxyl terminal direction. Remember, all these things have diagrams. They have structures rather, and I can't draw the structure here. I can't be drawing it here. So what we are reading is the notes, right? So yeah. So loops and bends as a heading. Roughly half of the residues in a typical globular protein reside in alpha helices and beta sheets, and half in loops, turns, bends, and other extended conformational features. Turns and bends refer to short segments of amino acid that join two units of secondary structure, such as two adjacent strands of an antiparallel beta sheet. A, a beta turn involve four amino acid residue, in which the first residue is hydrogen bonded to the fourth, resulting in a tight 180 degrees turn. Proline and glycine often are present in beta turns. Loops are regions that contain residue beyond the minimum number necessary to connect the adjacent regions of secondary structure. Irregular in conformation, loops nevertheless serve key biologic roles. For many enzymes, the loops that bridge the domain responsible for binding substrates often contain amino acid residue that participate in catalysis. Right? Now, helix motifs provide the oligonucleotide binding portion of DNA binding proteins such as repressor and transcription factors. Structural motifs such as the helix motif that are intermediate between secondary and tertiary structures are often termed supersecondary structures. Since many loops and bends reside on the surface of protein and are thus exposed to solvent, they constitute readily accessible sites or epitopes for recognition and binding of antibodies. While loops lack apparent structural regularity, they exist in a specific conformation stabilized through hydrogen bonding, short bridges, and hydrophobic interactions with other portions of the protein. However, not all portions of protein are necessarily ordered. Proteins may contain disordered regions, often at the extreme amino or carboxyl terminal, characterized by high conformational flexibility. In many instances, these disordered regions assume an ordered conformation upon binding of a ligand. This structural flexibility enables such regions to act as ligand control switch that affect protein structure and function. Now, as a heading, tertiary and quaternary structure. The term tertiary structure refers to the entire three-dimensional conformation of a polypeptide. It indicates in three-dimensional space how secondary structure features helices, sheets, bends, turns, and loops assemble to form domains, and how those domains relate spatially to one another. A domain is a section of a protein structure sufficient to perform a particular chemical or physical task, such as binding of a substrate or other ligand. Other domains may anchor a protein to a membrane or interact with a regulatory molecule that modulates its function. A small polypeptide such as triose phosphate isomerase, or myoglobin may consist of a single domain. Now, by contrast, protein kinases may contain two domains. Protein kinases catalyze the transfer of a phosphoric group from ATP to a peptide or protein. The amino terminal portion of the polypeptide, which is rich in beta sheets, binds ATP, while the carboxyl terminal domain, which is rich in alpha helix, binds the peptide or protein substrate. The group that catalyze phosphoryl transferase reside in a loop positioned at the interface of the two domains. In some cases, proteins are assembled from more than one polypeptide or protomer. Quaternary structure defines the polypeptide composition of a protein, and for an oligomeric protein, the spatial relationship between each subunit or protomers. Monomeric protein consists of a single polypeptide chains. Now, dimeric single polypeptide chain. Dimeric proteins contain two polypeptide chains. Homodimers contain two copies of the same polypeptide chain, while a heterodimer, the polypeptides differ. Greek letter alpha, beta, gamma, etc. are used to distinguish different subunits of heterooligomeric proteins, and subscript indicates the number of each subunit type. For example, alpha subscript 4 designates a homotetramic protein, and alpha subscript 2, beta subscript 2, gamma a protein with 5 subunits of 3 different types. Since even small proteins contain many thousands of atoms, depictions of protein structure that indicates the position of every atom are generally too complex to be readily interpreted. Simplified schematic diagrams thus are used to depict key features of a protein tertiary and quaternary structure. Now, ribbon diagrams, something is here, a diagram, trace the conformation of the polypeptide backbone with cylinders, an arrow indicating regions of alpha helix and beta sheet respectively. In an even simpler representations, line segments that link the alpha carbon indicate the path of the polypeptide backbone. These schematic diagrams often include the side chains of selected amino acids that emphasize specific structure-function relationship. As a heading, multiple factors stabilize tertiary and quaternary structure. Now, higher orders of protein structure are stabilized primarily and often exclusively by non-covalent interactions. Principal among these are hydrophobic interactions that dive most hydrophobic amino acid side chains into the interior of the protein, shielding them from water. Other significant contributors include hydrogen bonds and salt bridges between the carboxylates of aspartic and glutamic acid and the oppositely charged side chains of protonated lysyl, arginine, and histidine residues. While individually weak relative to a typical covalent bond of 80 to 120 kcal per mole, collectively, these numerous interactions confer a high degree of stability to the biologically functional conformation of a protein. Just as a Velcro fastener harnesses the cumulative strength of multiple plastic loops and hooks, some proteins contain covalent disulfide, that's SS, bonds that link the sulfhydryl groups of cysteine residue. Formation of disulfide bonds involve oxidation of the cysteine sulfhydryl groups and require oxygen. Intrapolypeptide disulfide bonds further enhance the stability of the folded conformation of a polypeptide, while interpolypeptide disulfide bond stabilizes the quaternary structure of certain oligomeric acids. As a heading, three-dimensional structure is determined by X-ray crystallography or by NMR spectroscopy. Now, X-ray crystallography. Since the determination of the three-dimensional structure of myoglobin, over 40 years ago, the three-dimensional structure of thousands of proteins have been determined by X-ray crystallography. The key to X-ray crystallography is the precipitation of a protein under condition in which it forms ordered crystals that diffract X-rays. This is generally accomplished by exposing small drops of the protein solution to various combinations of pH and precipitating agents such as salts and organic solids such as polyethylene glycol. A detailed three-dimensional structure of a protein can be constructed from its primary structure using the pattern by which it diffracts a beam of monochromatic X-rays, while the development of increasing capable computer-based tools has rendered the analysis of complex X-ray diffraction patterns increasingly facile. A major stumbling block remains the requirement of inducing highly purified samples of the protein of interest to crystallize. Several lines of evidence include the ability of some crystallized enzymes to catalyze chemical actions, indicate that vast majority of the structure is determined by crystallography. faithfully represent the structure of proteins in free solution. As a heading, nuclear magnetic resonance, that's NMR. Nuclear magnetic resonance NMR spectroscopy, a powerful complement to X-ray crystallography, measures the absorbance of radio frequency electromagnetic energy by certain atomic nuclei. NMR active isotopes of biologically relevant atoms include 1H superscript, 13 carbon 13 superscript, nitrogen 15, and potassium 31. The frequency of phosphorus, rather not potassium, phosphorus 31. The frequency or chemical shift at which a particular nucleus absorbs energy is a function of both the functional group within which it resides and the proximity of other NMR active nuclei. Two-dimensional NMR spectroscopy permits a three-dimensional representation of a protein to be constructed by determining the proximity of these nuclei to one another. NMR spectroscopy permits a three-dimensional representation of a protein to be constructed by determining the proximity of these nuclei to one another. NMR spectroscopy analyzes proteins in aqueous solution, obviating the need to form crystals. It thus is possible to observe changes in conformation that accompany ligand binding or catalysis using NMR spectroscopy. However, only the spectra of relatively small proteins less than or equal to 20 kilodalton in size can be analyzed with current technology. As a heading, molecular modeling. An increasingly useful adjunct to the empirical determination of the three-dimensional structure of protein is the use of computer technology for molecular modeling. The types of models created take two forms. In the first, the known three-dimensional structure of a protein is used as a template to build a model of probable structure of a homologous protein. In the second, the computer software is used to manipulate the static model provided by crystallography to explore how a protein's conformation might change when ligands are bound or when temperature, pH, or ionic strength is altered. Scientists also are examining the library of the available protein structures in an attempt to devise computer programs that can predict three-dimensional conformation of a protein directly from its primary sequence. As a heading, protein folding. The native conformation of a protein is thermodynamically favored. Now, the number of distinct combinations of phi and psi angles, specifically potential conformations of even a relatively small 15 kilodalton polypeptide, is unbelievably vast. Proteins are guided through this vast labyrinth of possibilities by thermodynamics. Since the biologically relevant or native conformation of a protein generally is that which is most energetically favored, knowledge of the native conformation is specified in the primary sequence. However, if one were to wait for a polypeptide to find its native conformation by random exploration of all possible conformations, the process will require billions of years to complete. Clearly, protein folding in cells takes place in a more orderly and guided fashion. Now, before we continue, I want to plug my power bank. I will remove this microphone because this is an iPhone. I'm using an iPhone XR to record this video. So that I can charge it a little bit because I'm on live. I'm using my power bank to charge it. Okay, so let's continue. Now, folding is modular. I hope you can hear me. I know the sound has changed a little bit. Let me put this off. Okay. Now, folding is modular. Protein folding generally occurs via a stepwise process. In the first stage, the newly synthesized polypeptide emerges from ribosomes and short segments fold into a secondary structural unit that provide local regions of organized structure. Folding is now reduced to the selection of an appropriate arrangement of this relatively small number of secondary structural elements. In this second stage, the forces that drive hydrophobic regions into the interior of the protein away from solvent drive the partially folded polypeptide into a molten globule, in which the modules of secondary structure rearrange to arrive the mature conformation of the protein. This process is orderly but not rigid. Considerable flexibility exists in the ways and in the order in which elements of a secondary structure can be rearranged. In general, each element of secondary or supersecondary structure facilitates
proper folding by directing the folding process toward the native conformation and away from unproductive alternatives. For oligomeric proteins, individual protomers tend to fold before they associate with other subunits. Now, as a heading, auxiliary proteins assist folding. Under appropriate condition, many proteins will spontaneously refold after being previously denatured. By treatment with acid or base, chiotropic agents or detergents. However, unlike the folding process in vivo, refolding under laboratory condition is a far slower process. Moreover, some proteins fail to spontaneously refold in vitro, often forming insoluble aggregates. Disorder complexes or unfolded or partially folded polypeptide held together by hydrophobic interactions. Aggregates represent unproductive dead ends in the folding process. Cells employ auxiliary proteins to speed the process of folding and to guide it towards the productive conclusion. As a heading, chaperones. Now, chaperone protein participates in the folding of over half of mammalian protein, the HSP70, that's 70 kilo Dalton heat shock protein. Family of chaperones bind short sequences of hydrophobic amino acids in newly synthesized polypeptides, shielding them from solvent. Chaperones prevent aggregation, thus providing an opportunity for the formation of appropriate secondary structural elements and their subsequent coalescence into a molten globule. The HSP60 family of chaperones, sometimes called chaperonins, differ in sequence and structure from HSP70 and its homologous. HSP60 acts later in the folding process, often together with an HSP70 chaperone. The central cavity of the donut-shaped HSP60 chaperone provide a sheltered environment in which a polypeptide can fold until all hydrophobic regions are buried in its interior, eliminating aggregation. Chaperone proteins can also rescue proteins that have become thermodynamically trapped in a misfolded dead end by unfolding hydrophobic regions and providing a second chance to fold productively. Now, as a heading, protein disulfide isomerase. Disulfide bonds between and within polypeptides stabilize tertiary and quaternary structure. However, disulfide bond formation is non-specific. Under oxidizing conditions, a given cysteine can form a disulfide bond with the SH of any accessible cysteine residue. By catalyzing disulfide exchange, the rupture of an SS bond and its reformation with a different partner cysteine protein, disulfide isomerase facilitate the formation of disulfide bonds that catalyze or that stabilize their native conformation. As a heading, proline cis-trans isomerase. All X-pros protein bonds where X represents any residue are synthesized in the trans configuration. However, of the X-pro bonds of the mature protein, approximately 6% are cis. The cis configuration is particularly common in beta tones. Isomerization from trans to cis is catalyzed by the enzyme proline cis trans isomerase. As a heading, several neurological diseases result from altered protein conformation. Okay. Prions. The first one is prions. Okay, I remember this prion. That is the time I made a video about prion proteins on my YouTube channel. So the transmissible spongiform encephalopathies or prion diseases are fatal neurodegenerative diseases characterized by spongiform changes, astrocytic gliomas, and neuronal loss resulting from the disposition of insoluble protein aggregates in neural cell. They include Creutzfeldt-Jakob disease in human, scrappy in sheep, and bovis spongiform encephalopathy, mad cow disease in cattle. Prion disease may manifest themselves as infectious, genetic, or sporadic disorders because no viral or bacterial gene encoding the pathologic prion protein could be identified. The source and mechanism of transmission of prion proteins long remained elusive. Today, it is believed that prion disease are protein conformation disease transmitted by altered function and hence the physical properties of protein. Endogenousness to the host. Human prion-related protein, the ROP, a glycoprotein, coded on short of chromosome 20, Normally, monomeric is a template for the combination transformation of non-reaching to salt. The extra complete polypeptide maturation. The best known defect in collagen biosynthesis is curvy, a result of a dietary deficiency of vitamin C required by prolyl and lysine hydrolysases. The result deficit in the number of hydroxyproline and hydroxylysine